The overall goal of this procedure is to determine the ability of ovarian cancer multicellular spheroids to invade a mesothelial monolayer. This is accomplished by first incubating red fluorescent protein-expressing ovarian cancer cells in low-adhesion culture plates to form multicellular spheroids. The next step is to plate green fluorescent protein-expressing mesothelial cells on fibronectin-coated glass-bottom culture dishes to form monolayers. The final step is to transfer the ovarian cancer spheroids from the low-adhesion plates to the plates containing the mesothelial monolayers. Ultimately, fluorescence time-lapse video microscopy is used to quantitate the relative ability of control and experimental ovarian cancer spheroids to form a hole in the mesothelial monolayer.
The main advantage of this technique over existing endpoint assays is that a combination of fluorescently labeled cells and time-lapse video microscopy is used to monitor the dynamics of mesothelial clearance over time.
Prior to the formation of ovarian cancer spheroids, it is necessary to prepare low-adhesion 96-well round-bottom culture dishes. To produce these culture dishes, add 30 microliters of polyhema solution to each well of a 96-well cell culture dish. Incubate the 96-well plates in a 37 degrees Celsius non-humidified incubator for 24 to 48 hours to evaporate the ethanol, leaving a film of polyhema on each well. This polyhema film prevents cells from attaching to the bottom of the well, forcing the cells to grow in suspension. The red fluorescent protein expressing ovarian cancer cells in this experiment are cultured in 10% base medium, a custom cell culture medium. When the low adhesion culture plates are ready, trypsinize a plate of ovarian cancer cells by first washing the plate with PBS, then adding 1 milliliter of trypsin and incubating the plate at 37 degrees Celsius for 5 minutes. After adding 10% base medium to quench the trypsin, transfer the cell suspension to a 15 milliliter conical polypropylene tube. Pellet the cells in a tabletop centrifuge at 900 RCF for 3 minutes. Aspirate the supernatant and resuspend cells in 10% base medium. After counting the cells using a hemocytometer, adjust the concentration of cells to 100 cells per 50 microliters of 10% base medium. Add 50 microliters of the uniformly suspended diluted cell suspension to each well of the 96-well polyhema-coated culture dish. Incubate the 96-well plate in a 37 degrees Celsius cell culture incubator for 16 hours to allow the ovarian cancer cells to cluster together, forming a single multicellular spheroid in each well. To begin this procedure, pre-coat the wells of a 6-well glass-bottom MatTek dish with fibronectin. Add 2 milliliters of fibronectin PBS solution to each well of the dish and incubate at room temperature for 30 minutes. The green fluorescent protein expressing mesothelial cells for forming a mesothelial cell monolayer are cultured in 10% base medium. Trypsinize a plate of mesothelial cells and spin down in a tabletop centrifuge at 900 RPM for 3 minutes. Aspirate the supernatant and resuspend cells in 10% base medium. Adjust to the desired concentration with 10% base medium. When the 30-minute fibronectin incubation of the MatTek dish is complete, wash the wells with 2 milliliters of PBS. Aspirate the PBS and plate 6 × 10 to the 5th mesothelial cells per well in each well of the 6-well MatTek dish. Incubate the MatTek dish in a 37 degrees Celsius cell culture incubator overnight to allow the mesothelial cells to attach to the dish and form a monolayer. To begin this assay, use a pipette to collect the ovarian cancer spheroids from the 96-well polyhema-coated plate. Aspirate the medium from one well of the 6-well MatTek dish containing a mesothelial cell monolayer. Wash once with 2 milliliters of PBS. Add all of the spheroids from the 96-well plate to 1 well of the MatTek dish. This is approximately 3 times the number of spheroids that are going to be imaged, to account for spheroids landing on the part of the dish that cannot be imaged.
Since we want to image only one spheroid per field of view, aggregation of the spheroids must be avoided. Rock the dish gently from side to side to distribute the spheroids evenly across the monolayer before they attach.
Place the MatTek dish on the stage of an inverted wide-field fluorescence microscope capable of performing time-lapse imaging for the duration of at least 8 hours. Use a motorized stage to image multiple positions in the dish with multiple spheroid intercalation events in a single experiment. The ovarian cancer cell spheroids will settle to the bottom of the dish and attach to the mesothelial cell monolayer. Collect GFP, RFP, and phase images of 20-plus spheroid monolayer interactions every 10 minutes for 8 hours. In the mesothelial cell clearance assay, the RFP-expressing ovarian cancer cell spheroids will invade the GFP-expressing mesothelial cell monolayer, creating holes in the monolayer. The sizes of the holes are measured by tracing the black holes in the GFP images using ECLIPSE software or another suitable software such as ImageJ. The size of the hole is then normalized to the initial spheroid size by dividing the size of the hole at 8 hours by the size of the spheroid in the corresponding RFP image at time 0. The mesothelial cell clearance assay can be used to compare the invasion ability of ovarian cancer cell spheroids that have been genetically modified to elucidate the molecular mechanisms by which ovarian cancer cell spheroids clear the mesothelial monolayer. In this example, the mesothelial clearance ability of OVCA433 ovarian cancer cell spheroids that have attenuated expression of TALEN1 was compared to that of control OVCA433 spheroids. The holes produced in the monolayer by the spreading spheroids was measured, and 6 positions from each group were averaged. As shown in this graph, the average clearance area created by TALEN1 knockdown spheroids was significantly smaller than the average area created by control spheroids, suggesting that TALEN is required for mesothelial clearance by OVCA433 ovarian cancer spheroids. This time-lapse movie shows a control OVCA433 red spheroid invading a green mesothelial monolayer. This next time-lapse movie is of an OVCA433 red spheroid with attenuated expression of TALEN1 invading into a green mesothelial monolayer. When compared with the previous movie, it is obvious that attenuation of TALEN1 expression in the spheroids decreases mesothelial clearance ability.
After watching this video, you should have a good understanding of how to monitor the interaction of ovarian cancer multicellular spheroids and mesothelial cell monolayers over time using time-lapse video microscopy.